Okay, hello. My name is Brian Mullan. I work for a contract development and manufacturing organization in the gene therapy area called Yposkesi. We're based in Paris, just south of Paris in Europe. I want to do a short presentation today around supply commercialization and associated challenges for gene therapy vectors. But I might take the opportunity just to present our company in a couple of slides beforehand, before I go into this topic. Yposkesi is, were founded in 2016. We were spun out from a research foundation called AFM Telethon, which is focusing on genomic solutions for treatment of rare diseases. And they have an associated foundation called Genethon, which should be well known to most of you. I think it's been working for the last 30 years in the whole gene therapy viral vector area. So we are a contract development and manufacturing organization focused on gene therapy products. So we have the full suite of, should we say, capabilities associated with this, process analytical development, industrialization for production, CGMP manufacturing, and we also can do evaluation of vector functionality. A part of our initial capabilities was in the cell therapy area, so we have quite a lot of capabilities in transduction, and also monitoring and estimation and measurement of gene expression from these vectors. There are a lot of CDMOs obviously in this area, so what's different about us? I think through our association with Genethon, as we effectively have access to a lot of the technology that they've developed over the previous years, has put us in, I think, in a strong position, certainly in two areas. We have a very high yielding process for AAV manufacturing, but we also have quite a strong process for suspension manufacturing of lentivirus vectors, which is becoming more and more prevalent these days. So as mentioned, we work with AAVs and lentis. We have two platforms. They both apply to each one, either adherent or suspension. The difference with these are we use cell factories or hyperstacks for adherent, but both of our adherent processes, and this is quite normal for the industry, contain serum. There's a certain level of scalability with these as well, in terms of the number of cell factories, of the number of hyperstacks that you can use, but this becomes somewhat limited depending on either your indication, if it's a muscular, or your foreseen market, if the needs would be quite high. So this is why we're seeing more and more clients moving towards suspension processes. We use these in single-use bioreactors. It's chemically defined media without serum, and this has much more scope for scalability for indications requiring high doses or quantities. So in terms of the overview of how the processes work, we start with a cell bank, we grow out the cells, and then we're using transient transfection processes, either a triple transfection for AAV or quadruple transfection for lentiviruses. We then run through purification, which can be 1 or 2 columns, depending on the needs, formulation, and fill and finish to make the drug product. Our process is largely single use, with the exception of certain chromatography resins, which can be reused as the resin has more than one cycle in its life cycle. Just to kind of recap it, to give an overview of all of that, in terms of our experience, we have quite a lot of experience gained since the inception of the company. We've made about 45 GMP batches for lentiviral vectors across 10 different transgenes, and about 40 for AAV GMP batches, also about across 10 transgenes. We also are working with quite a few clients on accelerated or fast-track projects, which I'll get onto when we talk about some of the challenges within the industry. So to briefly complete the circle, we have very well developed, I would say, capabilities for bioprocess development. We have a pilot lab which runs at the same scale with the same equipment into manufacturing. And within our manufacturing suite, we can run adherent processes or up to 2 by 200 liters for suspension, but this will soon increase to 2 by 1,000 and then 4 by 1,000 as we're investing in growing out the company, including adding a new building. Within fill and finish, we have 2 suites. For aseptic filling, we can do visual inspection and container integrity. And we are planning to increase our capacity here as well, also with the addition of an automated fill and finish line. With analytical development, I'll talk about this in 1 or 2 slides, but we have pretty much a full suite of methods that we can offer, including some of the vector functionality assays if this is of interest to people. And lastly, we also have a group on regulatory support who do regulatory surveillance and intelligence, and can also support clients with their filings for their investigational dossiers or interactions with health authorities. So this is my last slide before moving to the talk about basically challenges and a look ahead in the industry. So you can see an overview of a process between either AAVs or LV. This is reasonably standard. We have quite a lot of experience working with different transgenes at different scales. And you can see here that the scalability, I showed just for 2 transgenes here, one is an AAV8, another is an AAV9, is very, very scalable across from shake flasks up to small bioreactors up to 2 times 200. While I don't show the data for lentiviruses, we have basically similar scalability. And also with our models, the scale down models, they're very predictive of the next level up. So processes can be set up at 2 liters or 10 liters. And we have found through multiple projects that this helps us to set up the 50 and the 200 liters with quite a lot of reliability as well. So we can move through the various phases of development into provision of clinical lots quite quickly. So as you can see, we don't really see much of a loss of titer across these scales. While I show just 2 AAV types here, and I mentioned the LVs, we have experience with a lot of different AAV serotypes like 2, 5, and 6, and also capsid variants, which we're starting to see more and more. We can also pool to 200 liter bioreactors to give larger volumes until our 1,000 liter arrives. And part of this, I think, scalability and also the yields is due in part to a proprietary transfection reagent that we have called PTG1+, which gives stable transfection complexes, which are also stable in the reactors as well. So it means they have more time to interact with the cells. Just lastly on analytics, we have pretty much a full suite of analytical methods in-house that we can offer. We can also work with client methods, and we have a lot of experience either in plugging client methods into our analytical suite of methods, or transferring those methods in and finishing off the development with them. We also have a well-established network of external partners for specific tests, like for adventitious agents at bulk harvest testing. Okay. So moving on to the second, our main part of the talk, just talking a little bit about challenges and the evolving landscape for gene therapies. I think it would be fair to say that historically CDMOs worked to provide clinical material or tox material for clients. I don't think that would be quite the case going forward. And even today, we see more and more clients who are looking for their CDMO partner to be faster to market. They're working either with urgent needs or small patient populations with a view to commercialization. And we will see this in a slide or two. It is, I think, quite foreseen that some of these validation requirements, while ensuring safety and regulatory compliance as well, will have to be somewhat adaptable to try to get these medications to the patients who need them a little bit quicker. There's also the aspect of scalability as well. A lot of these are rare diseases that gene therapies are trying to treat, but not only. So if we invest in process development at a certain scale, we want to be sure that this can grow up as a product goes out onto the market. Lastly, with technology, I think someone once described that the gene therapy industry is somewhere similar to what the monoclonal antibodies industry was in the 1990s. I think monoclonal antibody manufacturing and development today is quite mature. And I think it is quite clear as well that gene therapy is going to evolve quite rapidly in the next 5 to 10 years to try to plug that gap and maybe come up to the level where monoclonal antibodies are today. And within this respect, there are some challenges for the industry. There are numerous, but I've just called out 4. Productivity, that we're, I think mainly due to the fact that we're working with transient transaction-based systems. The yields on the processes are quite low at 40%. I think with antibodies, this is towards 80. And I think the technology proposition today is good in some areas, but weak in others. Lastly, with lentiviral vectors, these are mainly used to transduce CAR-T type therapies today, but it is clear that lentiviruses as a product class in their own right will start to grow in the coming years. And I think there's an issue around overall stability, which will need to be addressed to enable that. So specifically, how we in Yposkesi can help if you're working on these type of products and you have some of these challenges in terms of speed to market. We have a lot of expertise and investing a lot in process development and characterization across multiple products. This has also helped us to work with some clients on plug and play approaches. And there's an initiative launched by the NIH recently called PAVE GT, which is also going in this sense, that we're trying to leverage development knowledge gained from other therapies onto new therapies, as opposed to doing them individually one at a time. And we're certainly starting to see this and propose this internally as well. We also are investing a lot in automation for analytical testing, which makes the test results come through quicker with more throughput and better robustness as well. In terms of adaptable process validation, typically for biologic or biotherapy, the process validation is a lot to do and it takes quite a long time. I think this will need to be made a little bit more flexible while ensuring the right balance of risk versus science and compliance. For us, even though we're not working on commercial manufacturing today, we are actually starting to develop already our validation strategies with documentation, hierarchies, and ongoing data generation. And this is to try to set a base for what a base case process validation package would look like, so that if we work with someone, we already are moving in this area, and if there's a need to move a bit faster, we have something that we can modify and adapt for the client needs. We're also applying analytical quality by design to some of our analytical methods to have more robust methods that we understand better. Just on the process validation point, if you take a look at what we call a standard approach for monoclonal antibodies, so for those of you who worked in validation before, you may see this differently. Some things may be missing, but I think it's a reasonably approximation of what you need to do. You can see various phases of clinical development, which the process and analytical development has to accompany, then going through the final qualification, the initial qualification, the submission and regulatory review, and then go into commercial supply. So there are various activities here that we as a company at the moment are starting to define strategies and approaches and capabilities for it to be ready when we need to work with our clients on these. But if you take this approach for monoclonal antibodies and apply it to ATMPs, you see basically that some timelines start to become very compressed because the phasing is not the same. Phase 1 and phase 2 can become phase 1 and phase 2 combined. Phase 3 is no longer pivotal. We talk about phase 2 pivotal. And we may have to do an initial qualification, submit, and then make some engagements and come back and do a later qualification on the process. So certain things will get compressed and other things will be extended out over time. In terms of scalability, I mentioned our suspension processes. These are very, very scalable, and I think would support scalability for future market growth. For technological evolution, we are actively doing, I would say, technology surveys, trying to identify where the industry is evolving and have innovation roadmaps in place which are proactively managed. We have partnerships with various industrial suppliers and academic partners, and we're also constantly evaluating new analytical technologies, such as robotization and various different formats of assays. Lastly, just on the challenges, the first two are somewhat linked, productivity and yield, but it's clear that injecting transient transfection complexes into suspension bioreactors and hope they meet the cell somewhere and express the virus, I think this approach is going to have to change. We are working with some external consortia in looking at new and novel approaches to try to increase the contact between the cells and the complex. And this is moving beyond novel transfection reagents or optimizing transfection reagents. Similarly, yield, overall the yield, there are various figures, but I think on average this is about 40% for some of these processes. I think there seems to be consensus, as far as I can see in the industry, that these need to be 10 to 100-fold higher at the end of cell culture, and then we can start to work on the downstream steps to try to improve the overall yield. Speaking of downstream steps, some resins are very well developed. There's some new resins for AAV which catch a lot of serotypes, less developed so for lentiviruses, but certainly if you need a second column, and given the levels of, say, DNA and host cell proteins we see in some of these columns, you may need a second column. Certain options there aren't so many, and I think the industry needs to invest in more of these resins. Lastly, with lentiviruses, which are becoming a product class in their own right, these are quite fragile. You can get a lot of loss during GP stability, and I think we need to better understand what's going on there, maybe at the formulation level, to try to increase the overall yields across the process. So thank you very much for for coming to see the talk, for your time. If you would like to talk to us a bit further, anything you saw today would interest you, please contact Patricia or Patrick, my colleagues, or myself. And if you would be interested in learning a little bit more about our company, we have a virtual tour which we can take you through, and it's basically an interactive video which we can accompany you through all the parts, development, QC, production of our company. So enjoy the rest of the conference, and thank you again for your time today.